So thank you especially Marco for the invitation today, and it's really a pleasure to be here. Here is my hospital from the top. It's a nice slide. It's impossible to read what is written here, so it's a little bit strange that I put this slide, but it's just something I wanted to show. So I am a neurologist, so I have a clinical activity, and then I have a research activity which is mainly dedicated to genetics, and I have a laboratory of genetics, of human genetics of neurological disorders, and my main interest is multiple sclerosis. So I decided to split my presentation into 4 different parts. In the first part, I will give you some definition on pharmacogenomics and personalized and precision medicine, which is something that probably some of you have been, let's say, exposed, especially in recent years. Then I will discuss pharmacogenomics in the context of multiple sclerosis, so in the context of an autoimmune or dysimmune disorder, let's say. And then I will give you 2 examples. One of a pharmacogenomic studies, a study in which we have been able to identify a genetic variant implicated in the response to one of the treatment of multiple sclerosis, which is Interferon-beta. And if I have time, I will also briefly show you an example of a transcriptomic approach, so a study in which you essentially measure mRNA levels using an unbiased approach. I will show you what means unbiased approach, trying to identify some biomarkers of response to treatment. So let's start with the most boring part of my presentation, which is definition, which I think are important to have in your mind. So we can talk as in terms of pharmacogenomics and pharmacogenetics. I think that it's important to divide the two definitions to distinguish. Pharmacogenomics is the study of variation of DNA and RNA characteristics as related to drug response, while we can define pharmacogenetics as a subset of pharmacogenomics, which is more implicated in the study of variation in DNA sequence as related to drug response. So this is a definition. I will make several errors during my presentation confounding pharmacogenomics and pharmacogenetics, but please try to remind this difference. So essentially, the basic principle is that you can study the impact of genetic code on response to treatment, but you can also measure the mRNA levels of several biomarkers as related to drug response. And of course, you can also use the so-called proteomics approach. So essentially, you measure protein levels in, in cells or serum or in general in biological materials, to see how it changes following drug administration. So there are these 3 levels, and I will give you an example of impact of DNA, of genetic variation on drug response to interferons, and then I will also give an example in terms of mRNA. I will not have time to touch the protein story, which is also very important. And how about personalized medicine? I think that it is a concept which is very much in use for clinicians, but I think that also basic scientists should be aware of it. So we can use a definition which is more complex. So we can say that personalized medicine is the tailoring of medical treatment to the individual characteristics, needs, and preferences of a patient during all stages of care, which include prevention, diagnosis, treatment, and follow-up. We can say that it is the capacity to provide the right drug at the right dose at the right time to the right patients, which is something which looks simple, but it's extremely difficult. And of course, in this issue, the use of genetic information is very, very important and relevant. Then we have precision medicine, which is a new concept. Maybe some of you are aware, in the United States, the President Barack Obama launched a very important initiative, which is called the Precision Medicine Initiative. And NIH put a lot of money in that, which is called the Precision Medicine Initiative. And the basic idea of precision medicine is that it is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. And this initiative, which is Precision Medicine Initiative, is very relevant from a public health point of view, and United States essentially made this launch this initiative far before Europe, and I think that Europe will follow probably in the next years, but we have to be ready for that. The basic idea of precision medicine, there are some reasons to launch precision medicine now, which is the fact that we have been able to sequence the human genome, so we have a large amount of data on potential biomarkers, including genetics. We have an improvement in technologies used for biomedical analysis, including all this bioinformatic approach using sequencing data and also integrating different layers of data. And we have new tools for using large data set, which is another very important element, to be able to integrate different layers of data and to be able to interrogate different databases at the same time. So the initiative has some near-term goals, which are mainly related to cancer. So the basic idea, and I will show why, precision medicine is now at a more advanced stage in cancer, for cancer treatment. So the basic idea is to be able to essentially design clinical trials of target drugs. So not the same drug to all the patients, but target drugs, so specific drug for a specific patient, the use of combination therapies, and also the capacity to overcome the issue of drug resistance, which is a problem of several treatments, especially in tumors. So you treat a patient, and then the patient becomes resistant, so the tumor is not anymore essentially changed and reduced by the drug that you're using. And then there are longer-term goals. And the basic idea is to create a cohort of more than 1 million American healthy controls on which to essentially be able to acquire several data, which include clinical data, data related to health and lifestyle, but also data in terms of genetics and transcriptomics and proteomics. So the basic idea to have a very large data set that could be used to better understand several aspects, including the impact of genetics on health, but also the impact of genetics in terms of drug response. What does it mean, personalized or precision medicine today? It can be useful to identify responder population, so to be able to provide the right drug to the right patients. And this is an example for breast cancer. So patients with tumor affecting the breast, overexpressing this gene, which is called HAR2, can be treated with this type of drug, which typically essentially decrease the expression of this particular protein. We also can use personalized and precision medicine to avoid adverse reactions, so to be able to know in advance which are the patients who will develop adverse events following a specific treatment. We will be able also to stratify patients based on the severity of their disease among high risk and low risk, and maybe to decide whether to treat patients with a high risk form of disease with a more aggressive drug, and patients with a less severe type of disorder with a less severe and strong drug. And then, of course, the basic idea is to reduce the trial and error prescribing. So to be able to choose a drug before the drug is tested and maybe before an error has been made, which is very relevant from a public health point of view. So the basic idea is that we have a population of patients with a given disease, a subset of patients is treated with a certain drug, and we have a smaller subset of patients who are actually responding to the drug. And this is interesting because it's a scenario which was, let's say, 15 years ago in multiple sclerosis. So we had a unique, we have two drugs at that time in multiple sclerosis, and as 30% of patients were able to respond for this specific drug. And the ideal goal would be to use in a smart way different drugs and to be able to provide to the right patients the right drug, and to essentially narrow down the number of patients who are not responders to treatment. So this is an example from a paper of last year. So just imagine that we have a drug with 22% of response. So it's not particularly exciting, but I think that in several diseases, the patients who actually respond to the treatment have this frequency. And of course, it depends on the definition that you use for response. This essentially corresponds to another measure which is used in epidemiology, which is called number needed to treat. So it means that you have to treat 5 patients to have 1 patient who actually respond to the treatment. Imagine to have a test, a serological test or measures done on a biological sample, and the test is negative in 70% of the population, and those who screen negative for this test have a probability of response to the drug which is 3%, which means that you need to treat 35 patients to have 1 who actually benefit from the drug. And imagine that 30% of the population who are positive for this test have a chance of 66% of response to treatment, which means that you have to treat 3 patients to have 2 who benefit from a disease. It is clear that if the drug is expensive and if the test is not particularly expensive, this makes a lot of sense to apply this test before to provide the drug. And this also allows to increase the efficacy, the overall efficacy of the drug, because we will be able to provide that drug only to the positive, to the patients who screen positive for this test and who have a very high benefit from this drug. So this is a very good example on how personalized medicine could help to better treat the patients. And genome-wide association studies are a way to identify essentially genetic variants associated with response to treatment. So just imagine to have a sample of patients treated with a specific drug. You have no responder patients, who are patients who do not have benefit from the drug, and you have responders patients who benefit from the drug. You divide and you create these 2 groups of patients, and then essentially you measure and compare the allelic frequency of several 100,000 of variants throughout the genome using the so-called array technology, which is mainly implemented by Illumina, and it is based on the typing of a particular set of SNPs, which are called tagging SNP. So you have to imagine that we know that there are several single nucleotide polymorphisms throughout the genome. So we are very similar, but we are also different. And most source of difference between individuals is based on single nucleotide polymorphism. So for example, in this case, it can be at this particular position in the genome, we can have a cytosine or thymidine nucleotide, and these essentially allow to categorize patients into CC patients, homozygote for the C allele, heterozygote individuals, so having 1 cytosine and 1 thymidine, and the third group of patients who are homozygote for the with a thymidine nucleotide base at this particular position in the genome. So the comparison between the frequency of these variants in responders compared to responders is essentially the basis of genome-wide association studies. Essentially, you look for variants which are differentially distributed, so which are more frequent in one group compared to the other. And then if you find something like that, you claim that there is a genetic association of a variant with the phenotype that you are studying. This is a Manhattan plot, and essentially each dot corresponds to a single nucleotide polymorphism, which is plotted along the x-axis in terms of its position along the chromosome. So that means that, for example, all of these dots refer to single nucleotide polymorphisms, which are present on chromosome 1 in different positions along the chromosome. And then dots are plotted along the y-axis, which is the negative logarithm base 10 of a p-value. So the higher is the number and the more extreme is the p-value, okay? So you have to imagine that you are typing 100,000 SNPs, and for each SNP you perform a statistical test, which is a chi-square. So for each variant, you compare the distribution, the frequency of the variant in one group compared to the other, and you use a chi-square test, and you have a p-value associated with each variant. So this means that you are performing 100,000 statistical tests. And you probably remember from statistical classes at university or even later, that if you perform several statistical tests, you need to adjust your cutoff p-value. So you're using 0.05 if you're using only a unique statistical test. If you are performing several statistical tests, you have to adjust your cutoff p-value and divide the cutoff p-value by the number of statistical tests that you have performed, which is called the Bonferroni correction. So to make the story short, this is the p-value which is Bonferroni corrected at genome-wide p-value. So you're testing more than 500,000 SNPs. What is beyond this cutoff p-value means that there is something which makes sense. What is below that, it can be noise, okay? So getting back to our example, so we have a drug which is effective in 30% of the population, meaning that you have to treat a little bit more than 3 patients who have 1 who benefit from the treatment. Just imagine to be lucky to find an allelic variant which is associated with drug response. 50% of the population carry these specific allelic variants. 50% of the population does not carry this specific allelic variant. And those who do not carry this specific variant have a probability of response to treatment which is 20%, meaning that you need to treat 5 patients to have 1 who benefit from the disease. And the group of patients carrying these specific allelic variants, which is associated with drug response, have a better clinical efficacy, which is 40%. So 40% of them respond to treatment, which means that you have to treat 2 patients to have 2 who benefit from the drug. This corresponds to a variant which is associated with an odds ratio of 2 of likelihood of response. So we can say that individuals who carry these variants have a probability to respond to treatment which is twice greater than those who do not carry that specific variant. And this is an example on how we can use genome-wide association studies for the identification of predictors of response to treatment. If there is something unclear, feel free to stop me, because I understand that these are kind of complex information, and maybe not all of you have this type of information. Is everything fine so far? Okay, so is personalized medicine a reality? I think that in oncology the answer is yes. This is a study of now 4 years ago. It's a very nice study, and you can see here that this is a, let's say, we can call a pipeline or a strategy of work in which essentially in less than 1 month it is possible essentially to perform a biopsy of a tumor, then to perform pathology, and then to sequence the tumor and to have information on the type of cells which essentially are part of a tumor. And this is a clinical application of this pipeline. These are 4 cases of patients with tumor which were refractory to conventional therapies, as you can see from here. Then, based on the results of sequencing in terms of tumor and using a combined genomic and transcriptomics information, it has been possible to identify that there were specific pathways which were, let's say, overexpressed and activated in these specific tumors, and to use drugs which typically target this type of pathways, and essentially to be effective in the treatment. So these are 4 cases in which the conclusion is that, and this is a typical conclusion from United States, in which you have to put everything in money, with $3,600 within 24 days, it had been possible to treat 4 tumors which were untreatable, which is, I think, a very good use of money for treating the patient. So this is an example of personalized medicine. So it has been possible to, by studying the specific individual characteristics of that tumor, it has been possible to provide the right drug and to essentially improve and treat the disease of a patient. How about multiple sclerosis? Okay, just a few words. Multiple sclerosis is an inflammatory and neurodegenerative chronic disease of the central nervous system, as previously mentioned by Professor Comey, it is characterized by demyelination, inflammation, and neuronal degeneration. It is a relatively rare disease. There are around 60,000 patients in Italy affected with multiple sclerosis. It is more common in women than men, with a ratio of 2 to 1, and it is a common cause of disability. Unfortunately, most of the patients develop the disease at young age, and 15 years after diagnosis, most of them have marked functional and cognitive deficit. It is an extremely heterogeneous disorder. Here you can see the autopsy of brain of different MS patients. You can see that most of the lesions are demyelinated lesions occurring around the lateral ventriculi, as you can see here. Some, especially in the first phases of the disease, there is the capacity of remyelination, so essentially to be able to essentially to compensate for the loss of myelin and to be able to recreate myelin in specific regions. The demyelinated lesions are also present in the cortex and in the deep gray matter. And you can see that each patient is different from the other, which makes more difficult also the issue of pharmacogenomics, because probably each patient has an individual story of a disease, and also probably each patient has an individual history of response to treatment. Of course, there are different clinical symptoms and signs, which goes from visual loss, balance problems, walking problems. So it's a very heterogeneous disorder also from a neurological point of view, and it is one of the best way to learn neurology for a resident in neurology. And here you can see the MRI. If you remember the previous slide showing MRI in the disorder related to perforin, here is different in the sense, I mean, it's similar because demyelinated lesions are diffuse. There is an asymmetry in most of the cases, but here you can appreciate in FLAIR images that there are hyperintense lesions occurring around lateral ventriculi and also, for example, in spinal cord. And this is the classical dozen fingers, which occur, which is demyelinated lesions perpendicular to the corpus callosum. So just to, this is not the topic, but just to give you an idea, this is the genetic architecture of multiple sclerosis. Each variant represents a risk factor for multiple sclerosis, and you can see that each variant is plotted along the x-axis in terms of their frequency in the general population. And here is the y-axis, which is the individual increase of risk provided by this specific allelic variant. And you can appreciate, since multiple sclerosis is a complex disease, when apart from HLA-DRB1 1501 and another marker which is very rare in the HLA region, all of the variants are very common in the general population, and they have a very small and modest individual risk. And I'm making this point because essentially, let me skip, okay, because this is a key issue, and I think it's very important to understand, also to understand the role of pharmacogenomics. The fact that sample size matters is very important. So this is a very self-explanatory. If you're looking for something for which your hypothesis is that it is something with a very large effect size, so if you're looking, for example, for a genetic variance and your hypothesis is that this genetic variance essentially is able to predict a response to treatment and have a very strong effect, you need relatively small numbers. So we're going in this direction of a graph. But let's say, unfortunately, in most of the cases, what you are looking for have a modest effect on the phenotype that you are studying. So for example, here, if you want to identify a pharmacogenomic variant with an effect of 1.5, so able to increase the risk to develop a disease of 1.5, you probably need something around 800 patients to study. While if you are looking for something with a modest effect on the response to treatment, you need a very large number, which is around 10,000 individuals, which means practically to type and to recruit 10,000, for example, MS patients treated with a specific drug, which you can imagine is a very, very large effort. And so going back to this part, okay, all this information have been achieved thanks to the fact that there was a collaborative effort putting together more than 10,000 affected cases in the first experiment, and then more than 15,000 in the second experiment. So the basic idea is that, especially in the field of genetics now, and also in the field of pharmacogenetics, if you just go by yourself, it's very, very unlikely to get any finding, unfortunately. And so you need to have collaborative efforts with other groups. So, as mentioned before, the basic hypothesis, one of the hypotheses related to multiple sclerosis, is the so-called outside-in model. So essentially, the basic idea is that self-reactive CD4 positive T cells, which are specific for certain components of myelin who are reacting against self, are able to reach the central nervous system. They probably cross the blood-brain barrier, where they are essentially reactivated by local antigen-presenting cells. And essentially an amplification of the immune response in the central nervous system leads to tissue injury. So this is the so-called inflammatory phase, and the hypothesis that this is the first phase of a disease, which is the phase of a disease which is better targeted by anti-inflammatory drugs. And then after this first phase, there is a second phase, which is a neurodegenerative phase, which is characterized by chronic inflammatory processes involving different agents, which then become compartmentalized behind an intact blood-brain barrier. So essentially, drugs work well in the first phase of a disease where inflammation is prominent and inflammation at periphery level, while in the second phase of a disease, unfortunately, it is too late to make changes. And this is reflected by the drugs which are now available. There are, different from the past, now to treat multiple sclerosis, there are several drugs available. So, for example, we have a set of drugs which is called rituximab and now ocrelizumab, which is another agent. They both act on B cells, on CD20 marker, which is expressed on B cells. Or, for example, daclizumab has been already mentioned, acting on NK cells. Or we have, for example, alemtuzumab, which is actually working on the surface molecule, which is called CD52, which is carried by T cells. Then we have other drugs like natalizumab, which is mainly working by essentially inhibiting the crossing of blood-brain barrier by activated immune T cells. And then we have some other drugs with a putative effect on the central nervous system. But the basic idea is that, or for example, we have fingolimod, which is acting by trapping lymphocyte within lymph nodes. So the basic idea here is that in a disease like MS, we have several different drugs. And so we have to decide which drugs to give to a patient. And this is very important because we have different drugs, as you can see from here. Some of them are called first-line drugs, and they are, as you can see here, very safe drugs, so no particular concern in terms of safety, but they are not particularly effective drugs, while we have second-line drugs, which are in the middle, and then third-line drugs, which are like natalizumab and alemtuzumab, which are very effective drugs, but they raise serious concern in terms of their safety. So if we want to make a decision, which is a clinical decision based on some elements, we need to design studies and to understand if there are some biomarkers which could help us to make this decision, which is sometimes very difficult because we have to decide with the patient if it is worth to choose a more aggressive drug or if we can stay on a safer drugs but less effective. And sometimes the decision is very hard. Of course, one of the way for making this decision is to look at the severity of a disease, which is the inflammatory activity in the patients. But there are also other information which need to be taken into account. And so if we want to just have a brief idea of the role of pharmacogenomics, let's start with Interferon-beta, which is the first drug used to treat multiple sclerosis. And so the mechanism of action of Interferon-beta is related to the binding with two receptors, which are called IFNAR1 and IFNAR2. And in case of binding of interferons, there is then the activation of a signaling cascade, which include the JAK1, STAT1 or STAT2, and then there is essentially the creation of a macromolecule complex, which include also another molecule which is called IRF9, which is then translocated from the cytoplasm to the nucleus. And then in the nucleus, it acts as a transcription factors, essentially binding to a particular sequence, nucleotide sequence, which are called interferon-stimulated response elements. And those genes which contain this specific sequence in the promoter, they are essentially upregulated following interferon administration. So interferon is provided, then there is the signaling cascade, and there is an upregulation of genes containing this specific sequence, which is called ISRE sequence. And of course, following this activation, there are several effects which are essentially protective against the disease. However, we know that the drug is effective in let's say something around 20–40% of patients. So the question is, if this is the mechanism of action, why only 20–40% of patients have benefit from this particular drug? And these are the different studies which have been done starting from the early 2000s to try to identify genetic variants associated with response to interferons. And as you can see from here, most of them were candidate gene approach. So what is a candidate gene approach? This means that you study the mechanism of action of a drug, and then you choose that particular molecules are more important in the mechanism of action of a drug. So for example, if NAR1 and if NAR2, it could make sense that some changes at the level of these genes could somehow explain a different response to the drug. Look at the numbers. So the numbers were between 100 and 200, so very underpowered study. And results were not conclusive. There was a lot of discrepancy in terms of results. Even if the mechanism of action and the choice of genes was correct. Then two genome-wide studies have been performed, and once again look at the number, which is 200, so once again relatively underpowered study. These two studies have been performed using a genome-wide association approach. So what is the main difference? Well, in a candidate gene study, you say, okay, if NAR1 and if NAR2 are interesting genes, so I'm taking polymorphism of IFNAR1 and IFNAR2, and I compare the frequency of these variants in responder and non-responders. So you are using an hypothesis, and then you follow what is behind your hypothesis. In genome-wide studies, you have to be, let's say, more stupid. You are not making any hypothesis before. You just say, I would like to have an unbiased view of the genome. I would like to screen the entire genome and try to see if there are some signals around. And this is the difference between genome-wide studies compared to candidate gene studies. The two studies I mentioned before, essentially they used a genome-wide approach. They found some SNPs which were associated with a response to the drug, which have then be replicated in another sample, which was, however, underpowered. And if you look at main results, you can see that, for example, there was two intronic variants at the level of a gene called GPC5, which were found in one of the study, while most significant results were completely different in the other study, pointing to a role of ion channel as potentially associated with response to interferons, once again an intronic variant. So results were different. And the two studies were important, but relatively underpowered. Then other studies more recently have been done. For example, this is a study of a Danish group, and essentially what they did was to type genetic variants, once again using a candidate gene approach, IRF5, IRF8, and GPC5, which is the glypican5 that you see before. And the basic idea was to compare the time to first relapse in MS patients treated with interferon. So to check whether there was a different efficacy of a drug, which was measured using another outcome measure, which is time to first relapse, in individuals carrying different allelic variants at the level of this gene. Once again, using a candidate gene approach. And the results were not significant. Another study more recently of a group of Comabella, including also us, found again another intronic variants at the level of another ion channel receptor and another SNPs at the level of another molecule implicated in mechanism of ubiquitination, found to be associated with response to multiple sclerosis. But once again, using a candidate gene approach. So we have a specific hypothesis in our mind, and we test specific polymorphism. And let me briefly describe in the last 15-20 minutes what we did in a project which was 7 years long. So a very, very long project, and a lot of work has been done, but I think it's a good example of pharmacogenomics. So, the paper was published in Annals of Neurology, and there was a very nice editorial on that. So, I mean, I don't want to promote my research, but I think it's a nice, I'm describing it today because I think it's a good example of a pharmacogenomic study which was, let's say, successful. So we started 7 years ago by collecting patients with a particular forms of multiple sclerosis, which is called relapsing remitting. They have to be naive patients, so patients treated with interferon as a first drug, okay? And we also asked for a treatment for at least 6 months with Interferon-beta. So the minimum treatment was 6 months. This is just to remind that if you are collaborating with clinicians, you have to check in a very, let's say, careful way how patients are selected, because it is very important that starting from biological samples, you are collaborating with people who actually put a lot of effort in selecting the right patients. And this was our inclusion criteria. We also had other exclusion criteria. I'm not explaining more of that. And what we did, since we had a relatively small number of patients, we decided to focus on the two extremes of response to treatment, okay? So we choose some criteria to be able to extract from the general population the two extremes of response to treatment. So we have responders patients on one side, which were patients with no clinical relapses and no new MRI lesions in the first 2 years of treatment. And then on the other side of the spectrum, we had no responders, which were patients with a mild decrease in the propensity to develop clinical relapses or develop several MRI lesions while on treatment. And this was important because the idea was to rule out the noise which is made by patients having a response which was somehow in the middle. So we wanted to study the two extremes of treatment, which is a very common design used, especially if you have a small sample size. And then we use the Illumina platform, which we have two scanners in Raffaele, so it has been typed in my laboratory. And what we found was that there was a lot of noise here, but we found a very strong signal of association, which was beyond the genome-wide level. So the magic number, which was somehow supporting the fact that it was a real effect. And this is a so-called regional association plot. So if we zoom into the region in which the variant is present in a gene called SLC9A9, which is a gene that if we, by using a candidate gene approach, we would have never chosen this type of gene. Okay, so that's the main advantage of an unbiased genome-wide approach. This was not among the candidate genes to be hypothesized as being able to influence the response to interferon. And here you can see the main signal and some other indirect signal of variants in LD with the original SNP, which is an indirect evidence of association. And interestingly enough, you can clearly see that the proportion of no responders, which are depicted in orange, increased for each additional copy of the G allele. Here you can see the odds ratio. So in our cohort of patients, which is around 160 patients, so a very small cohort of patients, we detected a signal because we found a very large effect size. So this is we found that being a carrier of these variants increased the risk of being a non-responders to the treatment of more than 5 times. So this is the reason why we found this variant as associated with such a small number of patients. And it was important also, by stratifying patients according to genotype, to look that there was an additive effect. So the more G allele carried and the higher is the likelihood of not responding to the treatment. And then we have to do what is very important in genome-wide studies. We have to replicate our findings in 3 additional cohorts of patients. And here you can see that the direction was the same. In a US population, we found an odds ratio around 2. And then we found a direction in the same effect, even if it was not significant at individual level. But if we combine the analysis, we found that there was an evidence of replication of a signal. This is very important for genome-wide studies. You need to replicate your findings in independent population. If you have a signal from your study, now it's not enough to publish it. You need to have a confirmation or replication of your signal, because unfortunately there is a lot of noise and there is a high risk of false positive signal. Here you can see the proportion of responders across the different cohorts tested, and you can see that there was a difference in terms of frequency of responders across the different population. You can see that the proportion of responders based on our data was higher than expected. And for example, you can see that the criteria were not exactly the same in the US population and in the France population, because we do not have MRI data in France and we do not have analyzed relapse rate in US. So that could explain why the signal was not exactly the same in the different population. And what we did was we found that this variant was associated with a better response, so a better phenotype in a particular group of patients. So we need to distinguish whether the signal was related to a real effect on drug efficacy or was related to the fact that individuals with that specific allele had a more severe forms of disease. Okay, you understand the difference. It could be also possible that individuals carry that specific G allele, they have a more severe phenotype of a disease independently of the use of a drug, which is one of the problems of pharmacogenomic studies. And this is the reason why we interrogated another cohort of patients treated with another drug, which is called glatiramer acetate. And we found indeed that there was not such a strong signal. So individual treated with another drug, there was no impact of this polymorphism on the response to treatment, suggesting that probably the effect was related to interferon response. So the variant was found to be here. Here is an intron. The gene is expressed in this direction. And here you can see that the SNP is contained in a linked disequilibrium block, which is fully contained in an intron of a gene. So we do not have a direct functional role of the variant. We are not dealing with, as mentioned before by Chris, a loss of function variant. We don't know, because this is an intronic variant, so it's not in a coding region of a gene. But we can hypothesize that there are some mechanisms which explain why individuals carry that specific variant have a different response to the treatment. So we started to look at the gene, which is a sodium hydrogen exchanger, which is localized, which is also called NHE9, and it is localized in the sorting recycling endosome. It's on the membrane, and it has an important role in regulating the intraluminar pH. So it is involved in the regulation of the pH of cytoplasm and on inside the endosome. It is highly expressed in immune cells, which is important because, of course, in case of genes which are not expressed in immune cells or in central nervous system, it is more difficult to think about a role on the disease. And it has been genetically associated with neurological disorders, mainly autism or other neurodevelopmental disorder, but not MS, at the time in which we started to study it. So the first thing that we did was to check whether individuals stratified by this particular genotype were different in terms of response to interferons. So essentially we collected healthy controls, we typed them, and we collected individuals who carry the variant, the genotype associated with a good response to treatment, with individuals with a genotype associated with a no response to treatment. And we extracted the PBNC, and we stimulated PBNC with Interferon-beta. And we then look whether there was an induction, so an upregulation of the gene following interferon administration. And we did not find any significant difference between the two groups, suggesting that there was a different, let's say, effect from interferon administration. However, we found that after providing interferon-beta, in PBNC there was a general upregulation of the expression of a gene. So essentially interferon was able to upregulate the expression of a gene at PBNC level, okay? Which makes sense. We also look at in GEO database, so we extracted some array data on clinical cohort present in the literature, and we found that indeed these are two cohorts of patients before starting interferons and during interferon treatment. And we found that there was a general upregulation, which was significant in both cases, following interferon administration. So what we found in our cell culture, PBNC culture providing interferon, was also found in vivo, or let's say ex vivo, in MS patients treated with interferon. So the administration of interferons tend to upregulate the level of the expression level of a gene in PBNC. We did also some bioinformatic analysis, and we found indeed that there are 4 different interferon-stimulated response elements, which are the ones that I mentioned before, which are present in the putative promoter region of a gene. And we somehow confirm that this is a gene which is influenced by interferon administration. So it belongs to the genes which are essentially induced by interferon, as previously mentioned to you. So even this makes sense. But that was interesting. We also found that, and this was consistent in untreated patients, but also in interferon-beta treated and in glatiramer acetate treated patients, that in general, individuals with a more active MS patient, so individuals with a more severe phenotype, tend to have a downregulation of a gene. So essentially, the gene was downregulated in individuals with a more severe form of disease. Okay, I will then summarize at the end of this piece of the story. So essentially, we did some functional work, which has been done in Boston by a collaborator of ours, which is Dr. Elia Mann at Brigham and Women's Hospital. Essentially, what we found was that there was a general downregulation of a gene starting 8 hours following differentiation of Th0 cells into Th1 and Th17 cells, which was not evident in Th0 cells, so in cells which were not differentiated, which somehow suggested that this gene may provide an inhibitory signal of T cell activation, because once it is highly expressed, there is no differentiation, while once there is differentiation, it tends to be downregulated. And by using a silencing approach, using shRNA, it was also possible to find that as you can see here, maybe it's here better to see, the frequency of CD4 positive interferon gamma producer cells were more highly induced in the SLC9A9 knockdown model. So once the gene was silenced, there was a higher frequency of CD4 positive interferon gamma positive cells, suggesting that the gene could be a negative regulator of interferon gamma signaling in T cell differentiation. And these are the different pieces of the story. We started from an unbiased approach and found an intronic variant at the level of a gene called SLC9A9 associated with no response to interferon-beta. We found that there was a general upregulation of the gene following stimulation with interferon-beta, which was found in vitro by our experiments, but also in vivo in MS patients. We also found that the same gene was downregulated in more active MS patients. And then the final piece of the story is that we found that the gene is downregulated during Th0 cells differentiation into Th1 and Th17, and it seems to be a suppressor of interferon gamma signaling during Th1 differentiation. So the basic conclusion that we put in the paper was that SLC9A9 is implicated in multiple sclerosis disease activity, and so we still do not know whether it is a signal specific of interferon response, or it is more related to the fact that individuals with these specific variants tend to have a less inflammatory role than individuals who carry the other variant. And this is important to mention because we are testing the same variant in patients treated with other drugs additional to glatiramer acetate to try to understand whether it is more related to prognosis or more related to response to treatment. And of course, probably this is one hypothesis, of course, but it is possible that what happened for the discovery of genetic factors associated to MS, this is a cartoon showing that in 2007 we knew of 2 genes as associated with multiple sclerosis, and now thanks to this scale of patients collected, we are now aware of the existence of now 200 genetic variants implicated in the risk of multiple sclerosis. The hypothesis is that for the response to treatment could be the same story, that we have been able to find a variant which is the one I have shown you, somehow implicated in response to treatment, and probably by increasing the number of patients in the short future, hopefully, it will be possible to identify additional genetic factors which are implicated in the response to treatment, which is now more difficult because there are several drugs, but this is one hypothesis. And this is the consortium which has been created for the study of genetics of multiple sclerosis, and there are some international initiatives which are, however, also very important. So the basic idea here is that it is more complicated because you need to have genetic information, but you also need a clinical database which follow longitudinal patients treated with certain drugs. But it is something that, being lucky to be funded, it could be solved in the future. We are also working on other follow-up. So we submitted, it is in press, a paper on the response, on the role of genetic variants of a response at 4 years to interferon. So once again, if we change criteria of response to treatment, or if we change follow-up time, also results can be different. And we are also testing a panel of replication of variants involved in the response to interferons. But of course, there is other works in response to other drugs, like, for example, glatiramar acetate, which is another drug I comment you about. This is a project done by the pharma company, and I think the data are not particularly exciting, but they are pharma companies, so they put a lot of money and they do not share results, which is one problem, I think. I think this is another way of looking at things. So the basic idea is that you develop, you test a panel of variants, and then based on the profiling of genetic profiling, you would be able to choose one drug or the other. And this is called the comparative pharmacogenetics, but I think it's a little bit early to say something about it. I think that one area of interest is the pharmacogenetics of adverse events, and I would be really very lucky maybe in the next year to be able to give a presentation on this part, because I think that it is a real problem now in MS to face. There are few patients who have very serious adverse events, and it will be very, very important to identify a priori those patients who develop these adverse events. And of course, one of the problem is related to sample size, because adverse events are very rare, so you need to have very large numbers, and it is somehow difficult. There are some interesting data on PML, which is a serious adverse event related to natalizumab treatment, and there is a group coordinated by David Brassat in France, who's actually collecting some interesting data, providing some biomarkers and genetic variants implicated in the risk of PML. But this is really a key point, and I'm quite sure that new data will be available in the short future. Okay, so let me conclude, because lunch is calling us. So, what we can state, and I'm using MS as a model, we can say that in MS, identified variants have a modest effect on the probability of response to treatment. At the present time, it's not possible to use in a clinical practice this type of variants to predict patients who respond or don't respond to treatment. And this is important to say to patients that we are not yet ready. I think that we need to invest money in that. And we need still to explore this issue, but there is no clinical utility so far. I'm not, promoting the variant on SLC99 as a way to decide which treatment to be given and offered to patients, because it is too early, and I don't think it's not fair to tell patients something which is not very scientifically proved. And of course, the future would be to try to develop composite markers, including genetic data, but also other, for example, biomarkers, like including mRNA, but also proteomics, or also including imaging biomarkers to have a better and to improve the accuracy of discrimination between responder and non-responders. Of course, as it was for SLC99, the interesting part of the story was not necessarily the clinical utility, but the fact that we identified a new mechanism related to immunological response and to essentially the MS phenotype, thanks to this type of approach, which could somehow help to better understand the disease, also to develop new treatments. And as I mentioned before, pharmacogenetics of adverse events and comparative pharmacogenetics are new fields of interest. And of course, the most important part, collaborative efforts are needed among clinical groups, but also between scientists having different expertise. I think that this is really, really crucial to try to collaborate in a fair way and to, expand further the field. Let me just acknowledge my group of people in San Raffaele and my different collaboration, and please feel free to make some questions to me. Thank you.